everyone and welcome back to our YouTube channel nerdybrains. So in our previous lecture we studied about the 3D double helix model of DNA which was proposed by Watson and Crick, and today we will be studying about types of DNA. So if you have missed our previous video, you can click on the I icon above. Now let's start the video. So here you can see I have drawn the table for the types of DNA. When we study about the types of DNA, we study about 3 DNA. One is the B DNA, one is A DNA, and one is Z DNA. So let's move on to these features of the DNA and discuss them one by one and discuss that how they make each DNA different. So the first one is seeing types of DNA. Types of helix. Types of helix का मतलब होता है कि let's suppose we have one DNA here. Fine. So the twisting of this DNA, like how this DNA is twisting, how the twisting is occurring, in which direction it is occurring, that is the types of helix. Like if it's rotating, it's twisting in the right-handed fashion, then it will be a B DNA. Then the A DNA also is right-handed, but the Z DNA will be left-handed, means the twisting will be left-handed. But in A and B DNA, the twisting is right-handed. Fine. The next feature says helical diameter. Helical diameter means this whole diameter of the DNA. Fine. So this is the helical diameter. Now in B DNA, it will be 2.37. A DNA, it will be 2.55. Z DNA, it will be 1.87. Means Z DNA का जो like the width and the helical diameter वो छोटा होगा. Fine. Rise per base pair. Now what do you mean by rise per base pair? This is a DNA structure, and this DNA structure will be having base pairs in them. Now each base pair, let's suppose this is one strand, this is second strand, and this is the base pairing occurring. Fine. So one base pair and this is the second base pair. Now between these two base pair there will be some space. This is a space which is present between two base pairs. This space is called as rise. This space, like what is the space between the space between two base pair? So this is known as rise. Fine. So rise per base pair means the distance between the two base pair in B DNA will be 0.34. In A DNA it will be 0.29, and in Z DNA it will be 0.37. Fine. So this was rise per base pair. Now comes distance per complete turn. Fine. So whenever we talk about distance per complete turn, this whole term is known as pitch. आपने सुना होगा questions में आ जाता है what is the pitch of this DNA. So pitch simply means distance per complete turn. Distance per complete turn का मतलब क्या होता है? Let's take this DNA. From one point to the next point is distance per base pair. So from here to here, this is one complete turn. This is one complete turn, and this distance is known as pitch. This distance is known as pitch. This is known as pitch. Okay, this is known as pitch. So this is the distance per complete turn. So if we look for BDNA, this distance is 3.4 in BDNA, 3.2 in ADNA, and 4.5 in ZDNA. Now if the distance is 4.5, तो इसका मतलब it is a stretch, because you see here, if the distance is 4.5 for ZDNA, then it means it's stretch, means distance ज़्यादा है, मतलब stretch हो रहा है. Fine. Now coming on to the fifth feature, that is number of base pair per complete turn. इसका क्या मतलब हुआ? अब एक complete turn में कितने base pairs present होते हैं वो बताने हैं. मतलब suppose this is the 1, this is 2, this is 3, this is 4, this is 5, 6, 7, 8, 9, 10. Fine. So this is what I am saying कि कितने बेस पेयर पर कंप्लीट टर्न में प्रेजेंट होंगे उसको कहेंगे हमारा नंबर ऑफ बेस पेयर्स, और वो है बी डीएनए में 10. सो बी डीएनए विल हैव 10 नंबर ऑफ बेस पेयर पर कंप्लीट टर्न, ए विल हैव 11 and Z will have 12. So this was number of base pair per complete turn. Fine. अब what is major groove and what is minor groove? See, whenever there is a twisting, तो उस ट्विस्टिंग के दौरान कुछ ग्रूव्स के फॉर्मेशन होंगी, कुछ बड़े होंगे, कुछ छोटे ग्रूव्स होते हैं. तो उन बड़े ग्रूव्स को, उन लार्ज ग्रूव्स को हम मेजर ग्रूव्स कहते हैं, एंड उन छोटे ग्रूव्स को, दीज आर द स्मॉलर ग्रूव्स, इनको हम माइनर कहते हैं. ओके, तो बड़े ग्रूव्स को मेजर एंड छोटे को माइनर बोलेंगे. जैसे कि इसमें भी यू कैन सी दीज आर मेजर एंड दीज आर minor. Similarly in here also and in here also. Fine. So major grooves जो हैं B DNA में wide and deep होंगे. Let's look it by the image. B DNA में wide and deep हैं major grooves. So यहाँ पे you can see this point, this one. So this is the major groove for BDNA, and this is wide and deep. You can see the spacing is more wide and it's more inside, so it will be deep. Then for ADNA, narrow and deep. Fine. Narrow means spacing कम होगी but deep होंगे. So this is less spacing but deep. Fine. So narrow and deep for the ADNA. Then for ZDNA, it's flat. You can see it's flat here. Here also. Fine. So it's flat. Now coming on to the minor groove, that is, it will be narrow and shallow. Like for BDNA, it is narrow, small, and shallow means it's not much deeper. It's very less deeper. So this is the minor groove. Fine. So for the BDNA, minor grooves are narrow, छोटे होंगे, and shallow मतलब deep ज़्यादा नहीं होंगे. Fine. Now coming on to the ADNA. In ADNA, minor grooves are broad and shallow. Broad means wide होंगे but shallow होंगे, मतलब डीप ज़्यादा नहीं होंगे, तो दे आर ब्रॉड बट नॉट मच डीप. ठीक है, सो ब्रॉडर बट नॉट मच डीप, सो ब्रॉड एंड शैलो, दीज आर द माइनर ग्रूव्स हियर. Fine. Now for the Z DNA, narrow and deep. Narrow and deep. This is the minor groove for Z DNA. Fine. So यहाँ आप देख रहे कि minor narrow है but deep है. Fine. They are deep but narrow. So yeah, these were the types of DNA which we discussed today. That's it for today, guys. Thank you for coming and joining in. I hope you guys have understood. If you liked the video, please hit the like button and share it with your friends. Also, if you have any doubts, you can ask them in the comment section below. Please subscribe and hit the bell icon so that you don't miss any of our updates. Thank you for coming. Bye bye. Have a great time.